Thanks, HUD. And I want to commend FASTSEB and the NIGMS for convening us today. And I'm really excited to be here to talk about a subject that is very dear to my heart, which is communication about biomedical research. So as HUD said, I've been a science journalist for almost 20 years. I started at Nature, where I worked for 15 years. Here in Washington, D.C., and I think I met Howard Garrison in my first year on that job, so it's awesome to see him here today again. As had said, I am now directing the Science Communication Program at the UC Santa Cruz, where what we do is train scientists to become journalists and communicators. And I can honestly say that I believe this is one of the most exciting and hopeful eras that science journalism has ever seen. Now, I recognize that the information ecosystem is more fragmented and more chaotic than it has ever been. And I realize that there is well-founded, broad concern about the level of societal discourse on all issues, including science. But I think that this changing landscape, while disorienting to scientific institutions and journalistic institutions alike, also could help lead to more direct and open and honest dialogue with sectors of society that have not been well served by science content in the past. So, great. In the course of my career, I've heard a lot of scientists lament the fact that we no longer have very broadly recognized public champions of science. And the name that I hear referenced a lot in these conversations is Carl Sagan, who is the astronomer who in the 1980s had a TV show named Cosmos that inspired millions of people around the world. And I can't tell you how many scientists have told me what we need is a Carl Sagan for their discipline, cell biology, biochemistry, physics, or the like. But it's becoming harder and harder for single individuals to have a broad impact across the culture. And I actually think that that can be a good thing, because the science communication landscape has become much more egalitarian and accessible to people across scientific disciplines and life stages. And we no longer have to rely on one individual to carry a message. And we no longer have to convince gatekeepers at a few nodes, such as at major broadcast networks or major newspapers, to distribute science content. So this means that our science journalism and communication can be more innovative, more diverse, more representative of a broader segment of the scientific community than it has ever been before. So instead of Carl Sagan, we now have a wide array of scientists and communicators who are shaping a bold new future for this field, including these women. So this is the graduating class this year from our program at the UC Santa Cruz. And these 9 women from a variety of backgrounds are part of the reason why I am so hopeful about the future of this field. So with that in mind, let's set the context for today's discussions. We are gathered here today to talk about responsible communication of basic biomedical research, enhancing awareness, and avoiding hype. So we all probably have our own ideas of what responsible communication looks like and what hype looks like. So I thought it would be helpful to set some baselines. So I'll start with hype. Here, our host for today, Judith Greenberg, has actually provided what I think is a really helpful definition of what hype looks like. She has said that hype is exaggerating the outcomes of research for whatever motives people have, leading to potential negative effects due to inaccurate portrayal of research. And what I like about this definition is that it encompasses the fact that all of the players in the journalism or science communication process can contribute to hype for various different reasons. The incentives for each of these groups are slightly different. And yet, engaging in this process can have negative impacts for each of the players in the hype cycle. So I thought it would be helpful to just show how this plays out in one example. So the example that I've chosen, and I want to stress that I'm not showing this example to point fingers. I'm really showing this example to illustrate how all of the players can come to be involved in, the hype cycle. So the example I've chosen is the situation of arsenic life. So this was a case a few years ago where researchers were studying bacteria from Mono Lake in California, including this researcher. Her name is Felisa Wolf-Simon. And the researchers thought that they had found an organism that could do this really astonishing thing, which was incorporate arsenic into its DNA. So that would have been a really groundbreaking result because arsenic is toxic to life. So this result was published in a very high-profile journal. The sponsoring agency issued a press release and held a press conference and said that the finding will alter biology textbooks and expand the scope of the search for life beyond Earth. Journalists ran with it. This particular story was in Nature, and Felisa Wolf-Simon herself got a lot of publicity for the findings. She was profiled in a huge range of publications, from Time magazine to Glamour magazine. The problem was that it all turned out to be wrong. So microbiologists studied the so-called arsenic life bacteria, and they found that while the bacteria were apparently able to absorb or ingest arsenic somehow, they weren't actually incorporating it into any of their biomolecules. So rather than using it to survive, it seemed like they were sort of able to survive despite nearly being poisoned, which is an interesting story, but not the story that was being told. So the microbiologists who refuted the original findings were able to publish their research. So you could say that this is an example where science did self-correct, except for in the process, journalists and the funding agency were pretty embarrassed, and the whole episode had a real negative effect on the young researcher involved, Felisa Wolf-Simon. So, for instance, she was asked to leave the lab where she was working when she published the original result. So this situation shows how all of the players in the science communication or the science journalism process can come to be involved in hype. So scientists need to publish splashy findings to advance their careers. Funding agencies want to be funding impactful research. Journalists need to gain an audience, and the science journals themselves are chasing the high-profile papers. And unfortunately, we can all get carried away, and when we do, it undermines public trust in all of our institutions. It's a real shame because nobody actually needs to oversell the results of research to get attention. So this is a paper that was published a few years ago, and to do this research, scientists examined all of the health-related press releases that were published in 2011 by 20 major UK universities. And the researchers looked at the original papers that the press releases were based on, and they looked at the news stories that were based on the press releases. And they found that about 40% of the press releases exaggerated the results of the original studies in some way, and that when the press releases exaggerated, the news coverage was also more likely to exaggerate the findings. But the most interesting result from this study to me was this, in the author's own words, that exaggerated press releases weren't statistically associated with greater news coverage, challenging the received wisdom that hype generates impact. So there's no real upside to engaging in hype, and there's a lot of potential downside. So hype is the problem. What is the solution? I think we can all agree that we want accurate communication, but how can we be accurate while still fulfilling our goals, whether it's to inform or to educate, maybe sometimes to entertain? In other words, we want to engage. How can we do that while still being accurate? And that's where I hope we can focus the majority of our discussion today. So I'd like to make 3 recommendations for responsible communication that engages audiences. And I want to point out that all of us in this room represent different interest groups in communicating science, but I think that we can all adapt these principles to our own goals and needs. So, what are these principles? The first is to tell a story. The second is to be authentic. And the third is to innovate. And now I want to show some examples of how we can use these principles to produce accurate and engaging science content. So first, tell a story. So one question that I often get from scientists, and that was alluded to earlier, is, how can I interest people in the process of science, not just the outcomes? I think both scientists and journalists alike can be really frustrated by the way that the science media landscape can focus on results instead of process. So, the public only hears about research when it hits a particular stage, when a finding has matured to the point where it's published in a journal, but they don't hear about the scientific process of developing hypotheses and testing and refining them. And they don't get that this is an ongoing cycle. And so scientists that I've talked to feel that this distorts the scientific process itself because it places a premium on sensationalized results, and also that the public doesn't understand the underlying process of refining hypotheses, and they just see the results of contradicting studies. So one solution to this conundrum is to tell a story that casts the process of doing research itself as the narrative arc. So I want to give an example. This is a story that I wrote in 2015 for Wired magazine, focusing on this epidemiologist, Lena Moses, and this is her in her lab in Sierra Leone. And when I interviewed her there in 2014, she had just embarked on a project with a bunch of other colleagues that was looking at the immunology of Ebola survivors. And this project had just gotten off the ground. The idea was that by studying the antibodies that Ebola survivors produce, researchers could gain some insight into how they are able to beat the virus, and that could lead to a lot of helpful things down the line. But obviously at the time that I talked to her, this project had just started, so there were no results to talk about. So how do you make an interesting story about this research? And the answer in this case was to tell the story of how Lena Moses and her coworkers conducted the research despite very difficult conditions in the middle of an Ebola epidemic in Sierra Leone. And so I'm going to read you a portion of the story. And to set this up, this is right after Lena Moses and a virologist named Robert Gary have collected blood from Ebola survivors, and they are about to process the blood to separate out the immune cells that make antibodies. Those are the things that they want to study. Moses's boss, a virologist named Robert Gary, separated the cells they needed from the blood, washed them, and added a pink buffering liquid to each tube. Gary printed the date and an ID number on each tube, and put them into a Mr. Frosty brand insulated container. Mr. Frosty, in turn, went into a portable freezer. Tucked safely inside, the samples chilled over the next 4 hours. It was crucial that they cooled slowly, so ice crystals wouldn't destroy the cells. Finally, at 11 that night, Moses and Gary donned purple disposable gloves, popped open the lid on Mr. Frosty, and loaded the little labeled tubes into metal cases cooled with liquid nitrogen. She handled each tube for no more than a few seconds. Even the tiny bit of heat from her fingers could warm the cells inside enough to kill them and destroy the knowledge they contained. She shut the case, ready for a journey to the United States. Past Ebola outbreaks killed as many as 90% of the people who got the disease. This most recent one did not, and nobody is sure why. Thousands of people got sick but didn't die. Their immune systems now make antibodies to the virus, proteins that can fight Ebola and win. Those antibodies are essentially the ideal medicine, or rather they would be if someone could unpack the biochemical manufacturing process that creates them. Those cells Moses had collected contained the key, the blueprint for making that hypothetical drug. So this passage describes the mechanics of conducting research presented in the context of a dramatic story about one day in a scientist's life. And you don't need to know anything about the results of this research to understand the stakes that are at play here. But not all scientists are working on something as immediately attention-grabbing as Ebola. So how do you interest the public in very basic research? Part of my answer to this question is to be authentic, to talk about what really got you as a scientist interested in the research in the first place, not what you have to tell your colleagues or your study sections to justify the research. I think a lot of pressure to hype findings comes from scientists or funders feeling that without tying research to something that is very relatable, they won't be able to interest the public in their science. And I do think it's a great idea to make science relatable, but I think that we should not forget the wow factor, the fact that you can engage people in being interested in science by tapping into their innate curiosity. So research does not have to be oriented towards making a drug for people to care about it. Just look at cosmology, where Carl Sagan and more recently people like Lisa Randall and Neil deGrasse Tyson are popularizing science that doesn't have a direct human health application. Instead, they are emphasizing the beauty of revelation. And as biologists, you certainly have that tool in your toolkit. You are studying systems that are as complex and as mysterious, maybe even more so, than the physical universe beyond this planet. And investigating this system is probably the grandest story ever told. This is the stuff of human drama, not of hype. And you shouldn't be afraid to access the wow factor that might have gotten you into science in the first place. So for instance, last year I interviewed this researcher. She's a structural biologist named Yang Yan, and she's using techniques like cryo-electron microscopy to resolve the structures of really difficult proteins. And I loved what she said about why she got into science. When I asked her about that, she talked about how as a girl, she had read a novel called Journey to the West, which features a character who's a monkey who has mastered the art of transformation. So here's what she said about that. As a girl, Yang Yan read a classic 16th century Chinese novel featuring a monkey that can transform into other animals. Yan wondered what it would be like to change herself. If you could shrink yourself into the size of a molecule or a protein, that would be a totally different world, she recalls thinking. Now, as a leading structural biologist, Yan inhabits that world every day, investigating the way proteins work at the level of atoms. It was almost destined that I would become a structural biologist, she says. Another example. Okay, where's my house? So in October, I participated in a live show put on by the Story Collider, which is this program that produces a podcast about science storytelling. And one of the other storytellers that night was Sebastian Alvarado. He's the top guy in this picture. And he's a biologist, and he studies epigenetics, or gene regulation that is not directly encoded in the genome. And at the time, he was actually working in the lab of Russ Fernald, who I believe is an NIGMS grantee. Sebastian talked about his lifelong love of comic books and how that got him into studying one particular epigenetic process, DNA methylation. When Sebastian started studying methylation, he was part of a lab that was looking at this in the context of human disease like cancer and depression. And he found that he was really not engaged by that line of research. So he started studying DNA methylation in ants. And in this clip, he talks about how he began studying ants and ties that back to his childhood love of comics, and more specifically one particular Marvel character called Ant-Man, who is able to drastically shrink and enlarge himself. So here, Sebastian is talking about how he started examining DNA methylation in worker and soldier ants.
A colony, the workers and the soldiers. So we picked up these larvae, we put them in a tube, we ground them up, we extracted DNA, and then we measured DNA methylation. And we actually started finding some really cool differences. We found that the workers were more methylated and the soldiers were less methylated. And we figured, well, if one is more and the other is less, maybe we could use drugs to manipulate this equilibrium. So what we did is we took these larvae, we put them onto a drop of drug, that would either increase methylation or decrease it. And we put a little tissue paper on top, and this tissue paper would kind of create this capillary force that would suck up the liquid and bathe these larvae. And we waited 2 or 3 weeks. At the end of those 2 or 3 weeks, we saw these ants emerge from their little cocoons, and it turned out that they became smaller and bigger, depending on our drug treatment. So we were shrinking the ants and enlarging them, which is pretty cool. In fact, the smaller ants were even tinier than exist in the colony we were picking the larvae from. So they were aberrant biology. They were so tiny that you can't even find them in the wild. That was pretty exciting. The first thing we did was obviously repeat this experiment with as many animals as we could and wait 2 more weeks. In those 2 weeks, I found myself coming into my lab, introduced, as I said, people who work with cancer, people who work with aggression and depression, and just walking up to them and saying, guess what? I'm Ant-Man. And
so what I really like about that clip is it's so relatable. It talks about the process of science, developing a hypothesis and testing it and running replications. And you can really sense his delight and excitement at doing pure biology and how that allows him to access the love of science that he felt in the first place. Finally, and this is somewhere that I think the current communication landscape is making our jobs easier rather than harder, we can innovate. And we have so many awesome tools at our disposal today, from animations to videos to podcasts to high-resolution images. And these are powerful tools to tell stories of biology. And I know that the NIGMS gets this because, you're investing in displaying high-resolution images outside of typical science venues. So for instance, where's my PowerPoint? In Dulles Airport and on Instagram. But you don't actually even need really powerful, expensive technologies to use digital media to tell engaging science stories. So, for instance, 2 of my students shot a video this year about circadian rhythms. And they wanted to tell the story of Carrie Parch, who is a researcher at UC Santa Cruz who's studying the molecular clock complex, which is the series of proteins that control circadian rhythm. And they shot a video about this, and the whole video is shot in the format of what we call a whiteboard explainer, which is basically a time-lapse videos of pictures and diagrams drawn on a whiteboard. So I'll just play a little snippet of this.
It's also called clock. When we say molecular clock or just clock, we are referring to all of the proteins together. Circadian rhythm researchers like Carrie Parch at UC Santa Cruz are looking at these proteins to see if they can find a way to reset the molecular clock when it gets confused. Researchers knew that one of the proteins, called Cry, had a big influence in controlling the molecular clock, but they weren't sure how. In a recent study, Parch figured it out. It turns out Cry docks to the molecular clock through a small pocket. An arm reaches out of the protein clock and latches onto Cry through the small pocket. She is now looking for small molecules that could snuggle into the pocket and reset the entire molecular clock.
So this is shot on an iPhone using the most basic production equipment of a whiteboard and dry erase markers. Of course, if you have more resources, you can do more. So this is something on maybe the opposite end of that spectrum of fewer to more resources. This is a clip from a show called Science Scope that one of our alums developed for PBS NewsHour. And in this clip, Sikon Akpan is talking with a fluid mechanics engineer named John Cromaldi, who is studying how smells move in air.
It has a high degree of structure. Smells are tiny chemical molecules that get swept around by turbulent air. But these odors can also travel underwater, where creatures like fish and hermit crabs can smell them too.
And it turns out that the physics of flows in air and flows in water are exactly the same.
So Cromaldi's team measures these odors underwater. They start by pumping 5,000 gallons of water through a 50-foot-long tank. Next, they spread laser light into a sheet that slices through the water, and then finally comes the dye.
And so what we're doing is we're using a special kind of dye as a surrogate for the odor. And this dye has exactly the same types of properties that an odor would have if it were in this flow. But it has a special property that if we shine a laser light on it, then it fluoresces.
What they find is odor moves like taffy. It's constantly being pulled and stretched into thin filaments. If you've ever caught a whiff of a bakery when you walk down the street, you've picked up a filament. In some areas, odors get dragged away, leaving a blank space. This blank space is called intermittency. Small disturbances like a quick sniff are the tugs in this smelly tug of war. Here's a demonstration built by Cromaldi's team. A tube is standing in for an animal's nostril. So now breathe in. Now exhale. And you create an olfactory cyclone that changes the structure of the odor plume. So whenever you're in a garden and you stop to smell the roses, just remember that every sniff you make is changing the aroma for someone else. Next time.
So this was one of the inaugural pieces in the Science Scope series, which CCON developed for NewsHour, and this piece won one of our field's most prestigious prizes last year, the Kavli Prize in Science Journalism. So I hope I've given you some examples of how telling stories and being authentic and embracing innovation can help us do more accurate and engaging science communication. Now, I realize that we face a lot of challenges. So one of the questions I've been thinking about a lot is how can we take these tools and principles and apply them in ways that broaden the reach of science communication beyond people who have typically sought it out in the past. And to that end, I'm really excited to be able to announce today that we are one of 20 projects that has just been awarded funds from the John S. and James L. Knight Foundation to study ways to combat misinformation and promote public trust in journalism. And our particular project is going to focus on ways to use digital and social media to do more innovative and engaging science communication. And we have some really awesome partners on this. Our pilot is going to be in climate change, and we would love to branch out into other areas of research. Another problem for the field is that if we're aiming to reach all sectors of society, our field itself does not at the moment represent all sectors. So this is a problem and something that I'm really committed to working on at UC Santa Cruz in our science communication program. One of the issues that I'm encountering or that I'm hearing about a lot is that there's no robust sources of funding for students who want to do the kind of training that my program or a handful of other programs across the country offer. So that's a heavy lift for really the best candidates for this kind of training who have often invested many years and resources in gaining a PhD or a master's or something in between. And it's hard for them to, accept the idea of taking on debt to make a career change. But the good news is we have a lot of ideas and a growing pool of talented people who are laying the foundations for a more effective and inclusive science journalism. And I look forward to a productive discussion about how to move these ideas forward. Thank you.